Hello dear students, I am Dr. Iram Khan. Today we will start the unit on biotechnology for class 12th. In the earlier episodes related to this unit, we have already seen centrifugation and ultracentrifugation, which are the techniques used in biotechnology. Dear students, this unit on biotechnology is having two chapters, which are Biotechnology Principles and Processes and Biotechnology and its Applications. We will start with the chapter on Principles and Processes of Biotechnology. For convenience, I have divided this chapter into three parts. In the first part, we will discuss about the principles of biotechnology. In the second part, we will focus on tools of recombinant DNA technology. In the third part, the discussion will be focused on processes of recombinant DNA technology. Students, ever since the days of French philosopher René Descartes, who was a renowned mathematician and biologist of 17th century, all human knowledge, especially natural sciences, were directed to develop technologies which contribute to make the life of human beings comfortable. All the approaches to understand natural phenomena became anthropocentric or human oriented. The subjects like physics and chemistry gave rise to engineering, technologies and industries, which all worked for human comfort and welfare. Students, as we know that the major utility of the biological world is as a source of food. In this regard, biotechnology, the 20th century offshoot of modern biology, changed our daily life. Here, it is essential to take the name of Herbert W. Boyer, born in 1936. He is a researcher and entrepreneur in biotechnology. Along with Stanley and Cohen and Paul Berg, he discovered a method to coax bacteria into producing foreign proteins, which was the initial point for the field of genetic engineering. And we can say that this breakthrough was the basis upon which the discipline of biotechnology was founded. Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to human beings. Here, we should not confuse biotechnology with the processes like making curd, bread or wine, because they all are microbe-mediated processes. Today, the term biotechnology is used in a restricted sense to refer to such of those processes which use genetically modified organisms to produce the same on a larger scale. There are many other processes or techniques which are also included under biotechnology. For example, in vitro fertilization, leading to development of a test tube baby, synthesizing a gene and using it further, developing a DNA vaccine, correcting a defective gene. In this way, a proper definition of biotechnology is very important. The European Federation of Biotechnology, or EFB, has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. According to EFB, biotechnology is the integration of natural science and organisms, cells, parts thereof and molecular analogues for products and services. Dear students, now let us discuss about the principles of biotechnology. There are many techniques of biotechnology. Among those, the two core techniques that enabled birth of modern biotechnology are genetic engineering and maintenance of sterile ambience. Here, genetic engineering is a technique which is used to alter the chemistry of genetic material, which are DNA and RNA, to introduce these materials into the host organisms in order to change the phenotype of the host organism. Maintenance of sterile or microbial contamination free ambience is necessary in genetic engineering processes. because it enables growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. Dear students, we are aware of the advantages of sexual reproduction over asexual reproduction. The sexual reproduction provides opportunities for variations and formulation of unique combinations of genetic setup, some of which may be beneficial to the concerned organism as well as to the population. On the other hand, asexual reproduction preserves the genetic information. Traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. The techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allow us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. Dear students, let us examine the likely fate of a piece of DNA which is somehow transferred into an alien organism. Most likely, this piece of DNA would not be able to multiply itself in the progeny cells of the organism. But when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because of the fact that the alien piece of DNA has become part of a chromosome which has the ability to replicate. Students, in a chromosome there is a specific DNA sequence which is called the origin of replication. This sequence is actually responsible for initiating replication. Therefore, for the multiplication of any alien piece of DNA in an organism, the DNA needs to be a part of a chromosome which has a specific sequence known as origin of replication. This is why alien DNA is linked with the origin of replication, and as a result this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid of Salmonella typhimurium. Here, let me tell you, the plasmid is an autonomously replicating circular extrachromosomal DNA. Here in this illustration, you can observe a bacterium with enclosed plasmids showing chromosomal DNA and plasmids. It should be mentioned that Stanley Cohen and Herbert Boyer accomplished this process of artificial recombinant DNA molecule formation in 1972. They did this by isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at specific locations became possible with the discovery of the so-called molecular scissors or the restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. These plasmid DNA act as vectors to transfer the piece of DNA attached to it. Students, as you know that mosquito acts as an insect vector to transfer the malaria parasite into human body. In the same way, a plasmid can be used as vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistant gene with the plasmid vector became possible with the enzyme DNA ligase, which acts on cut DNA molecules and joins their ends. This makes a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA. When this DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host's DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant gene in Escherichia coli was called cloning of antibiotic resistant gene in Escherichia coli. In this way, we can make inference that there are three basic steps in genetically modifying an organism. The first is identification of DNA with desirable genes. Second is introduction of the identified DNA into the host. Third is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. So students, let us conclude the points discussed today. We have looked into the meaning, definition and advent of biotechnology. We have also discussed genetic engineering, which includes creation of recombinant DNA, use of gene cloning and gene transfer, and maintenance of sterile ambience as the two core techniques of biotechnology. We also inferred to the basic steps of genetically modifying an organism. In the next episode of this chapter, we will discuss the various tools of recombinant DNA technology. Thank you.